Hey everyone, my name is Riggs Rivera from your Bio 3150 class or your microbiology class. And my Bacteriophiles podcast was about plant promoters producing polymers. So, in this podcast, the host, Jesse North, talks about how plants benefit from the polymers produced by bacteria. The plant promoter in this case is the bacteria Herbosporillum ceropedicae, so right here. Herbosporum ceropedicae is a spiral-shaped bacterium. It is also an endophytic bacterium, which means that it is usually found on the surface of plants, but also can be inside the plant. And these types of bacteria, despite being near or inside the plant, do not cause any harm to the plant. Its genus Herbosporum is a group of bacteria that helps with the nitrogen fixation process with plants. So, just as a review, in this process, the bacteria helps convert the nitrogen in the air into ammonia, and the plants then take the ammonia and synthesize them into organic compounds containing nitrogen, such as amino acids, proteins, and nucleotides. So Herbosperm ceropedicae also establishes a symbiotic relationship with a variety of plants, and these plants are like rice, corn, sugarcane, and for this podcast, they focused on a certain species of grass called Setaria viridis, or it is also known as the green fox-tailed grass. So besides assisting in the nitrogen fixation process with plants, Herbosporum ceropedicae also produces a special polymer called polyhydroxyl alkanoate, or PHA. So similar to how our bodies produce fat to store energy, the bacteria will store extra nutrients and carbon in the form of these polymers in their cytoplasm. So interesting fact about PHA is that unlike fat, it's pretty solid, so it can be extracted and turned into bioplastic. But for the bacteria, and similar to how our bodies use fat, the bacteria can use PHA as a source of energy at times where nutrients isn't as available. So the specific type of PHA that Herbosperm ceropedicae produces is called polyhydroxyl butyrate, or PHB. Since there is barely any information on PHB's role on plant growth, scientists from the Applied and Environmental Microbiology wanted to figure out if PHB was responsible for the growth of plants that Herbosperm ceropedicae was symbiotic with. So again, it's commonly symbiotic with corn, rice, sugarcane, but for this study, they decided to focus on the fox-tailed green fox-tailed grass. The reason why they chose this plant was because it was a C4 model plant, and C4 is a certain type of carbon fixation that this plant undergoes, where, long story short, it turns carbon into sugars. And if you remember, PHB stores carbon, so they wanted to see if that exchange helped in the plant growth. To test this, the scientists took the genome sequence of Herbosporum ceropedicae strain SMR1, and this is a normal functioning strain. So they determined that this sequence had 13 genes that were involved in the metabolism of PHB. With this sequence, they were able to construct a variety of mutant strains that altered the regulation, synthesis, and degradation of PHB, as well as the flagella production. The roots of the fox-tailed plants were inoculated for 25 days with these strains. So the results are shown here. So figure A right here shows how SMR1 and the mutant strains affected the total area of the green fox-tailed grass. The control plants right here are those that were not inoculated. Again, SMR1 is a normal strain. PHAC1 through PHAR right here have defects in PHB production. PHAZ 1.2 have defects in their mobilization, and FLIA was classified as a mutant that did not colonize the plants at all. The reason why it did not colonize was because it was a mutant strain that did not synthesize flagella. So comparing PHAZ1 and 2, this mutant strain had limited movement, whereas FLIA did not have any flagella, so it had no movement. So as expected, the normal strain had the most total root area, and the mutant strains really not so much, especially the ones that had a problem with their movements. They're kind of low on this side. The scientists conducting the experiment calculated that SMR1 increased the total root area by 37% in comparison to the control. So next is figure B, which showed the number of lateral roots the plant produced. As you can see, the results were very similar to figure A, where SMR1 had the most lateral roots produced, and with the mutant strains not really producing as many. Next is figure C. So figure C shows the root weight of the plants, and these results show that there was not much difference between the strains and the controls, but interestingly enough, if you notice, PHAP2 is that its results are nearly identical to the normal strain, and this is because PHAP2 is a wild type of strain, which means it has normal function. So the scientists also fused the strains with green fluorescent proteins, or GFP, and this fusion gave the scientists an image of the rate at which the bacteria formed microcolonies in the intercellular spaces of the plant roots. So for the image A and B, the strain that was fused was PHAC1, which again was one of the mutant strains that had a defect in PHB production. These images show the microcolonies, which are being pointed by the red arrow here. And these colonies were forming as early as 4 days. Images C and D show PHAZ1, which again was the mutant strain that had problems with its movement. And these images show that the microcolonies began forming at around 10 days. The last set of images, E and F, was with PHAZ2, which again had problems with its movement. So similar to C and D, these microcolonies did not start forming until the 10th day. So if you compare images A and B to that of C, D, E, and F, it shows that the microcolonies in A and B formed much faster than in C, D, E, and F. Their explanation for the results was that PHB synthesized early when the colonies were formed and mobilized much later, so the bacteria can utilize the stored carbon. And if you compare this figure with the previous figure, you'll notice not only is PHB production and regulation important, but mobilization is also important. So the scientists concluded that PHB production of Herbosporum serrapedicae contributed to the growth of the roots of the green fox-tailed grass, with Herbosporum serrapedicae also being symbiotic with many staple foods. Studies like this could lead to advancements in food production and other agricultural advancements. Remember that PHB could also be used as bioplastics, so as we gain more and more information about PHB, and as we understand it better, we may find more future uses for it. Thank you.